Yes, I
Yes,
am
he
here.
is there. Yes, you also been made co-host.
Yeah.
Thank you so much for joining us. And on behalf of Kausalya, the Skill University, Department of Skill Development, Government of Gujarat, I welcome you in this faculty development program,
Thank you.
which is mainly based on the agri innovation, agri
Mhm.
biotech
Okay.
entrepreneurship. And for last 6 days, we have been talking about various types of agri innovations, market scenario, various components like plant agriculture, biofertilizer, biopesticides, and innovation management technology transfer aspect.
Okay.
Today, the last day, we are covering hydroponics, second agriculture, what we're going to talk about. And we'll have perspective from the commercial aspect from the Kumar Forest, Pune, on
Mhm.
the floriculture, tisculture aspect. So I welcome you, Dr. Singh. For the information of audience, Professor Singh is working in Gujarat Biotechnology University as a professor. Also, I think you have taken charge of Dean Research. It seems from your profile. Before that, Professor Singh was working with one of the very important DBT institute in Mohali. Yesterday, we had a very good presentation from Dr. Tiwari also. So, before that, we had a very good interaction on that IP part, technology management. And he is very leading expert in various kind of secondary agriculture product, functional sugars, and many more. And this is kind of translation research he is doing. Whatever project, product he is developing through various R&D project, it is worth commercializing and lots of industrial application. Professor Singh is fellow of various society, INSA fellowship, NASI, this National Academy of Agriculture Sciences. Large number of publication, 12 patent already granted. So list will go very long. Uh, so thank you, Professor Singh, for accepting our invitation to give this presentation. I welcome you and over to you. Yeah, you can share your slide. You have been given post, right?
Yeah, so let me share my slides. Okay. How to share? I have to learn. Just a minute. Yeah. Share the screen, I
Yes,
guess.
share the screen. Then yes, started coming. And you have to just make it in a slideshow mode. Then done. Yes, perfect.
Fine. So I hope I am audible to all.
Yeah, yeah, perfect.
Fine. So, as the topic allotted to me, I am supposed to speak about around value-added products and secondary agriculture. So, I will be sharing some of innovations which our group has made in last 8 to 9 years. So, we have done some of the catalytic innovation for bioproduction of value-added products and secondary agriculture. At present, I am working at Gujarat Biotechnology University, which is located in near the gift city at Gandhinagar, Gujarat. So, a couple of slides I have made just to introduce our university. It is my responsibility to introduce Gujarat Biotech University. It is the first university created on this specific subject, biotechnology, and it is considered to be the brainchild of our Honorable Prime Minister Modi, and it has been established through Gujarat Act 16 of 2018. It comes under Department of Science and Technology. We have five major departments at Gujarat Biotechnology: medical, animal, plant, environment, and also industrial biotechnology. So I am currently at Industrial Biotechnology. This is the leaders of our university. The vice chancellor is called as Director General, and he is Dr. Subir Majumdar. He happened to be a great one of the leading scientists of India. He has already founded one DBT institution, NIAB in Hyderabad. He is fellow of all three academies. FNA, FASC, FNASC, and he's also JC Bose Fellow, and he got Tata Innovation Fellowship also from DBT. Then we have in hierarchy, we have a director. He happened to be director of ICGB Kolkata before joining GBU. So he's also fellow of two national academies. Then we have 5 departments which are headed by different eminent scientists like Professor Rawal, Dr. Vishal, Senator Sangram, Chirayu, and myself. So this industrial biotechnology department specifically, if I tell, because my work is mostly on industrial biotechnology, but yes, pertains to the agriculture, secondary agriculture work. So this department, the main aim is to generate human resource ready for industry. So we train our students not only for lab scale processing, but also we train our students for process engineering to upscale the process. And also we give a sense of entrepreneurship. We give a sense of startup incubation and how technology is developed and how scale up facilities are developed. So basically, we also give a sense of market knowledge to means we also feel that students should also know the market knowledge of the technology which is being developed at GBU, and also they should know the operational work for the time and resource management, etc., which are required for industrial development. So these our faculty is working on different aspects like biomass valorization, carbon capture, metabolic engine, process engineering, bioinformatics aspect. We have a very good bioinformatics support in our department. Then the most important thing for any department is ideation. So many ideations of our scientists, our faculty is being appreciated by sponsoring different sponsoring agency, DBT, DST, NRF now, and GSBTM also, which is part of government of Gujarat. This is the complete strength of our faculty. We have 4 very bright scientists designated as assistant professors and 2 very senior scientists as associate professors, and they have most of the faculty are having very good exposure of abroad laboratories and very good fellowships also from abroad, like JSKS fellowship, very good fellowship from Japan. Then we have Marie Curie fellow, like Sandeep is a Marie Curie fellow. We have then Ramalinga fellow, like Ravindra and Sudhir both are having Ramalinga fellowship and Ramanuj fellowships also. So these are very brilliant group of scientists at Industrial Biotechnology Department at GBU. So now I come to the statement of problem, which my work actually is related. And basically, I am a molecular biologist by training, but my research group is working on the aspect of cell agriculture, cellular agriculture, or secondary agriculture. But yes, this problem is directly or indirectly related to this statement of problem, that is related to obesity and increasing incidences of diabetes in India. We all know that diabetes is becoming the capital of means India is becoming the capital of diabetes, and also obesity is one of the major issue. And this issue is not only related to our tradition to have more added sugar, to have sugar sweetened products, but also we are being trained to take the highly processed food products. In fact, there was a report published recently in Hindu, and it was recorded that there is a significant decrease in the sale of sugar sweetened and highly processed product in high income countries. So those to compensate this loss, we are being targeted by the industry. The low income countries or middle income countries are being targeted by these industries, and we are being trained to take pizza, to take highly processed product, to take, for example, Coca-Cola, too much sweetened products. So these all are leading to become obese. These are leading to increasing the incidence of obesity and diabetes. So what are the alternatives available today? So alternatives are different sweet molecules which are artificial sweeteners, but there are side effects related to associated with these artificial sweeteners. They have also undesirable taste. They cannot be used at bulk sugar. What is bulk sugar? We need to understand that when we purchase any sweetened product like kaju ki barfi, if we purchase, so lot of sugar it contains apart from kaju. So the amount of sugar is more than the kaju, which is for which it is actually being sold. So therefore these sweeteners cannot be used as bulk sugar. I am not sure that how these people are selling the sugar-free sweets. So how they are managing this is a matter of discussion. So now this slide shows what the overall contribution my laboratory has done as of now. So the biomass sugars of low value, for example, sucrose, glucose, fructose, the table sugar. So these are sugars which are readily available and they are of low value. So my group in last about 10 years, we have worked on generating novel enzyme variants with superior catalytic properties as demanded by the industry for catalytic transformation of these sugar molecules into high value function molecules, for example, allulose, trehalose, tauranose, allose, epilactose, trehalose, and also the starch can be converted to resistant starch. So these all molecules are having anti-diabetic, anti-obesity potential, not only anti-diabetic, anti-obesity potential, but also they exert prebiotic benefits to human health, and several other health benefits are associated with these molecules. So therefore they are called as very good pharmaceutical importance. But also these molecules have importance in agricultural research also. So on these molecules, some of our I will be sharing some of our contribution on how catalytic innovations, how innovations in biocatalyst systems can generate these kind of molecules. And these the star which I have put in these molecules, this shows that it is first report from India, from our laboratory has made in India. So these are the molecules which are required to be biomanufactured, which is the main soul of at present's policy of Indian government. So these D-allulose, T-hallose, and they all are having emerging market, but unfortunately there is no manufacturer in India. So for example, D-allulose, if we say, D-allulose is a carbon third epimer of fructose. At carbon third position, you can see the location of hydroxyl group is reverse. So this D-allulose is not recognizable by our system. So therefore, our system do not metabolize it, or very least metabolism is reported about D-allulose. So it gives low glycemic impact. So if we consume it, it gives a glucose suppressive impact. It gives an anti-dyslipidemic impact, means a lipid accumulation is reduced. Neuroprotective impact is also recorded by different laboratory. It gives antioxidant property. It is a very good molecule for decreasing adiposity. And so it has been proven that D-allulose is a good candidate for synbiotic. Synbiotic means a mixture of prebiotic as well as probiotic is called as synbiotic. So use of D-allulose for making any product increases the stability of that product, increases the texture of food products, increases, it has a better water holding capacity. So it gives a better gelling capacity to the food material. So consumption of D-allulose gives a better mouth feel. And most importantly, our intestine system can tolerate D-allulose. For example, a person of 60 kg can consume 54 gram of D-allulose per day, means 0.9 gram per kg of body weight can be consumed, and it has been accorded GRAS status by as far as FDA and FSSAI approval is concerned. So these are the global manufacturers of D-allulose. It is being available in US, Japan, Korea. China is a major producer of D-allulose now, but there is no manufacturer in India as of now. And what is the hurdle? The poor thermal stability of enzyme is considered as a major hurdle in industrial production of D-allulose. So when I started working on D-allulose in a DBT laboratory in 2015, so we thought to increase thermal stability of one enzyme, and this enzyme comes from agrobacterial tubefaciens. It is called as D-cyclose 3-epimerase or D-allulose 3-epimerase. It has cyclose and allulose are basically its new name is allulose, its old name is cyclose. So we introduced a stable tag called as SMT3, and introduction of this tag increased its thermal stability by several fold, by about 16 fold. So its half-life of wild type enzyme was 63 minutes, and the improved the fusion, this fusion protein exhibited the half-life of 943 minutes. About 16 fold improvement in half-life was observed. And the most important thing was that it was not the stability in the improvement in stability was not compromised, what not at the cost of its activity. The KCAT, if you see, the this fusion protein even showing a better KCAT than the wild type enzyme. So means the improvement was not at the cost of its functionality of this enzyme, which was most important thing. Then we applied this D-allulose 3 epimerase containing cells or the enzyme itself in different juices, and we observed that D-allulose can be formed, which was confirmed by different analytic analysis. And we also could see that the fruit and vegetable processing residues, they contain a lot of fructose which can be converted into D-allulose, and we have seen that vegetable waste is waste, and also the apple pomace type of waste could generate D-allulose or D-cyclose from this waste, and this was confirmed by TLC and HPLC analysis that yes, we can generate waste to wealth, but it involves a lot of purification cost. Therefore, we propose that we should use purified form of fructose for making the allulose. Otherwise, this enzyme is capable enough in generating wealth from the waste, the fructose available in different wastes. So this was done. This was our previous work, which was done by using a known enzyme. It was a patented enzyme, and it was modified by using molecular biology tools. Now we thought to explore Indian bioresource. We thought to explore different ecological niches available in Sikkim Himalayas, available in Tatapani, Chhattisgarh geothermal field, available in copper mines. So these cold lakes, these hot springs, these acidic uh, ecological niches, our team visited at different places, and we collected samples, and we extracted total DNA, and we did in-depth meta taxonomic profiling, in-depth metabolic potential analysis by using bioinformatic tools, and we published these in-depth studies in different international journals. So this is, for example, now this method could explore not only the cultivable resource available in the biosphere of these ecological niches. So for that, actually, I collaborated with one microbiologist expert, Dr. Amit Rai, at present working at Navi, and we together explored a lot of cultivable microbial resource, useful microbial resource, which can be used for different processing steps. But we know that about 97 to 98% resource is uncultivable. So that uncultivable microbiota can be addressed by extracting total DNA and doing functional metagenomic screening by cloning of different DNA fragments and by using molecular biology tool, by using recombinant methods of expression. So we explored this as a molecular biologist. Our group contributed in exploring novel genes which were possibly encoding enzymes of industrial importance. So, for example, this D-allulose epimerase enzyme, this gene was discovered from a thermal spring of 95 degrees Celsius, and when we sequenced this gene, we found no match at nucleotide level and also at protein level. And when we expressed this gene, we found that its stability was very high. Its thermal stability was very high. In fact, it was the most thermostable protein known so far. So it was showing about 165 hour of half-life at 60 degrees Celsius and 55 hour of half-life at 70 degrees Celsius. It again, this high stability was not compromised. Its turnover number means the ability to transform D fructose to D-allulose per second. So it was the first D-allulose epimerase reported from India. Then in this sequence, because when we discussed with several industry, or the industry suggested that yes, this is a very stable enzyme, but at 60 degrees Celsius, its activity is 70%, about 70%. So they said that please discover enzyme which should be active at 100% at the temperature of 50 to 60 degrees Celsius. So its catalytic activity should not be reduced at lower temperature. Although it is very good that it is a highly thermostable, highly thermal active also. but they suggested that just look for another variant. So we looked for another variant, and it was showing very high activity at 50 degrees Celsius. You can see the activity profile, that it was showing high activity at 50 to 60 degrees Celsius temperature, and its half-life was also excessively high, with 600 of half-life at 50 degrees Celsius. Without compromising kcat, you can see more than 300 kcat per second. We could find it, and you can see the half-life was about a month at 50 degrees Celsius. So it was liked by industry, and they are working on it. They have licensed this invention. Then industry always becomes greedy. Industry says that now can you look for one enzyme that can be functional at room temperature. So we recently came up with one variant of DLO-CF epomerase. It is functional from 4 degrees Celsius to 80 degrees Celsius. So we are working at present on in-depth characterization of this enzyme. So this is a comparison. When you do research, you have to compare it what is available in the world. So you can see that these are the different variants of DLO-CF epomerase reported from different laboratory, from different part of world, but none from India as of now. And our laboratory, in blue color, we have contributed some of the variant of DLO-CF epomerase, and you can, if you compare, our stability is far better than the previously reported enzymes, and without compromising KCAT. For example, if you see this version of enzyme shows 1853 minutes of half-life at 50 degrees Celsius, but its kcat is 68, and here you can see the half-life at 50 degrees Celsius 36,000 minute, and its kcat is quite high. So means without compromising the catalytic function, the catalytic activity of enzyme, we could achieve higher stability of this enzyme. But there was an industrial issue when we discussed with different industry. So we found that we hardly have a fructose producing industry. So therefore, we realized that we should also have indigenous invention for producing fructose. So we again mined our metagenomic data resource, which we have generated from by exploring different thermal ecological niches, and we could find that this glucose isomerase, which was able to transform glucose to fructose, and it was again a very stable enzyme. And now by using this glucose isomerase or xylose isomerase and deallose by using the synergetic action of these two enzymes, we could produce deallose directly from D-glucose. So this was also appreciated. This invention was also appreciated by one industry, and this is a comparison of biochemical properties of xylose isomerase or glucose isomerase, which could generate fructose from glucose. Then one of means our group member, they were interested in allose. Allose is transformed version of allulose, and this allose is having anti-cancerous property. It is a pharmaceutical molecule, but very costly to be used in food system, because it is yet to get FDA approval, FSI approval for its use in food. But yes, it has approval for pharmaceutical uses, and it has anti-tumor, anti-herpestive, cryoprotective, and these all roles are very appreciative lore. And in our laboratory, we are able to produce allose from allulose, but it is very costly to be commercialized. And yes, this invention is a contribution of scientific contribution from our group. This is a comparison on the allose telomerase. So this trehalose is another very important molecule, which is not only in high demand of in food industry, but also in pharmaceutical industry and cosmetic industry. Food industry, it is important molecule because it can reduce freezing point of food product. So it avoids formation of ice crystal. So it protects the protein. It protects it. Therefore, it increases the shelf life of food product. So therefore, it is very important in food. Most of the frozen products, they use trehalose. For example, the ice creams, it is a need to use trehalose. Then in pharmaceutical industry, it is important molecule because it stabilizes the cell membrane and the proteins. So it is used for cryopreservation of sperm cells and blood cells. In cosmetic industry, it is important molecule because it retains moisture. So most of the cosmetic products, the moisturizers, they contain trehalose to retain moisture. So it is a very important molecule for all useful in all three industries: food industry, pharmaceutical industry, and cosmetic industry. Unfortunately, again, there is no manufacturer of trehalose in India. So we mined our data and we could identify one gene which was encoding trehalose synthase, and it is a very important enzyme that can transform maltose to trehalose by using a glycosyl transferase reaction. For example, maltose is having it is comprised of two glucose molecules linked with alpha 1,4 bonds. So it first hydrolyzes and rejoin it. So by alpha 1,1 bond, now it is called as trehalose. So likewise, it was having a dual catalytic property because it can also act on sucrose and it can hydrolyze and rejoin. So it is a glycosyl transferase reaction, and it is called as trehalulose, another important rare sugar molecule. So it was having capability of dual catalysis at a time. So it is an important enzyme. Again, it was because it was discovered from a highly thermophilic environment, a very uh, uh, high temperature aquatic habitat. So it was showing a very good half-life, about 96 hour of half-life at 60 degrees Celsius. So again, it is a very thermostable trehalosynthase, and in fact, it is the most thermostable trehalosynthase reported so far. And most importantly, it is active in a wide range of temperature from 5 degrees Celsius to 45 degrees Celsius temperature. At present, we are working on this on the upscaling of this technology at GBU on trehalosynthase, and this is the biochemical comparison of trehalosynthase. You can see that there many trehalosyntheses are reported. Some of them are characterized for trehalose yield also. So our yield is a good yield, we substantially good yield we could obtain. For example, 66% conversion, 74% conversion at low temperature 5 degrees Celsius, and without means its stability was also very high. You can see its stability is again unmatched with other variants reported so far from different part of world, but none from India. It is the first report to the best of our knowledge on trehalosynthase. Then our group also worked on turanose. It is a natural isomer of sucrose. So this amylosucrose can again, by using glycosyl transferase reaction, it can transform sucrose into turanose by rejoining this glucose and fructose molecule by via alpha 1,3 bond. Now this turanose is a useful enzyme because it is, and these all are natural sugar. These are present in different natural products. For example, honey contains turanose, allulose, and these natural sugars, but are present in rare in traces. So our as a molecular biologist, as a industrial biotechnologist now, our duty is to develop processes for upscaling or for high yield of turanose by using biocatalytic production. So it also gives it also reduces consumption of turanose is also reported to reduce the lipid accumulation in our body. It is a low glycemic index sugar. So but yes, again, the FDA approval is yet to be obtained, and FSI approval is yet to be obtained. So therefore, we call it an alternative sweetener of next generation. But it has uses in pharmaceutical industry because it is used for some. I, I need to recall it that in some pathology, some pathology, it is important molecule. So by exploring the metagenome of one hot water habitat, we could discover one gene, and it was successfully making turanose from the table sugar, the cheap table sugar, 25-30 rupees per kg available in the market, and we could generate turanose, and by using analytical technique, we could confirm that yes, it is turanose which we are making. Now, this is a comparison of, so our enzyme is, yeah, I'll not say that it is the best, but it is one of the best enzyme for Tyranose production. And the most important one, very good thing was that this amylosucrase enzyme can also be useful for making alpha arbutin. by using hydroquinone as a substrate. Its glycosylation of hydroquinone makes alpha-arbutin. Alpha-arbutin is a useful uh, product because it inhibits tyrosinase activity. Now, this tyrosinase activity, inhibition of tyrosinase activity reduces the melanin production. So the cosmetic products, because we have been ruled by the white people. So in India, it is tendency to become white. So there are creams available which claim to reduce melanin production, and thus they make you fair. So so it has a great demand in India, but again there is no manufacturer of arbutin. Actually, we love to miss our Indian system, our Indian industry. We love to procure these kind of molecule from China. So without doing any effort, like we procure twice. We have lot of kids in India, but our kids are they hardly play with Indian toys. So what we do, we just procure it from China. So there is hardly any means very less manufacturer of toys, Indian toys. Likewise, these molecules our industries are procuring from China. We don't have any therefore we don't have innovation also. So probably our laboratory for the first time reported a catalytic method for production of arbutin. So a state like Gujarat, which is basically very bright, which has a bright future in terms of industrial development. So I am means Dr. Shukla and all with the help of all, I am encouraging the industries to come and take up these innovations from our laboratory, help us in making us understand that what more improvement, scientific improvement they need. We are ready to do it. So these are the estimated global market for alpha-arbutin. Again, there is no manufacturer of India in India, and we have this innovation in our hand to get it commercialized. So this is a comparison of D-amylosaccharides for D for sorry for arbutin production, and our enzyme has done well in terms of performance for arbutin production. Likewise, a very abundant molecule in Earth is starch, so which is a carbohydrate. So, but yes, starch has the consumption of starch again is a problem. So, our group was interested to convert this starch in some useful molecules. So, as I explained, that starch is one of the most abundant polymer, a carbohydrate polymer, a very important source of carbohydrate. Starch Starch is mainly made up of amylose and amylopectin. And so 20 to 30% amylose and 70 to 80% amylopectin are present in starch. So in amylose is comprised of glucan chains, which has glucose linked via alpha 1,4 bonds. In case of amylopectin, it is branched glucan chains, and these branches of glucan chains are attached via alpha 1,6 bonds. So there are different starch hydrolyzing enzymes. For example, alpha amylase, it hydrolyzes starch into maltose glucose. Beta amylase, it hydrolyzes starch into maltose. Glucoamylase, it hydrolyzes maltose into dextrin and dextrin into glucose. Then isoamylase enzyme, it specifically cleaves alpha 1,6 linkages found in amylopectin and glycogen. Type 1 pulinase, it also acts at alpha 1,6 bond. For example, here it will act and it will convert amylopectin to amylose. Pulinase type 2 enzyme, it acts both at alpha 1,6 and alpha 1,4 and converts starch into glucose. So, starch hydrolyzing enzymes are useful not only in food industry for baking purposes, but also in paper industry. Starch hydrolyzing enzyme are useful to make it more fine, more printable, to increase its strength. Also in textile industry, also starch hydrolyzing enzyme are useful. In pharmaceutical industry, starch is useful for increasing bioavailability of some active ingredients. In biofuel industry, starch is starch hydrolyzing enzymes are very important for bioproduction of ethanol and bioethanol. So when we consume starch, our intestinal enzyme convert starch into glucose. So there should be a starch which should not be hydrolyzed into glucose. It is in favor of our gut health, it is in favor of our overall health. So that starch which cannot be metabolized or less having less digestion or resistant to digestion is called as resistant starch. There are some natural forms of resistant starch, for example legumes, whole grains, tubers. They have some resistant starch, and on that basis resistant starch is divided into different types. For example, type 1 resistant starch. Type 1 resistant starch is a resistant starch which is not accessible to be digested for digestion for digestive enzyme. So these all these RS1 type or resistant starch type 1, these are found in grains, seeds, and legumes, and it is actually entrapped in some non-digestible material. So it is not physically inaccessible to digestive enzyme. Likewise, RS2 or resistant starch type 2 enzyme, it is found in unripe banana and raw potatoes, and it has granular starch. So the granules are resistant to, if you take as such granule, if therefore unripe banana is in Ayurveda, it is preferred to take because it is having resistant starch. So granular starch, it is having resistant starch. So what is the beauty of resistant starch? It is not digestible to our system and it reaches to gut. So when it reaches to gut microflora, they ferment this resistant starch because they are able to ferment it. And when they ferment it, they produce short chain fatty acid and several other metabolites that have several known and unknown functions. Likewise, RS3 type 3 resistant starch, it is uh, prepared by some enzymatic and physical method, which I will be explaining in my future slides, in my coming slides. And there is type 4 resistant starch, which is made prepared after chemical modification. This I don't cover because I am biologist. Then there is type 5 resistant starch, which is made by complexation of lipid molecules. This I will be covering in my presentation. So resistant starch is important for our digestive health. It can also control our blood sugar level. It is important for weight management. It reduces the chronic diseases like type 2 diabetes, heart disease, colon cancer disease, and also these are different studies that have proven that consumption of resistant starch is important not only for avoiding the type 2 diabetes and obesity, but also several other important health benefits it exerts. So, resistant starch type 3, if we consider it is prepared by using type 1 pullase enzyme. Type 1 pullase enzyme acts on alpha 1,6 bond and it converts it converts amylose amylopectin to amylose. For example, this is amylopectin. So when these branches are attached via alpha 1,6 bonds and type 1 pulinase acts on alpha 1,6 linkages, so it these branched structure converts into linear structure, linear chains, linear glucan chains. So, but type 2 I have not used because it chops both alpha 1,4 and alpha 1,6 linkages and generates maltotriose, maltopentose, and maltose. So, application of pulinase, it shows that it has several industrial applications, but at present I am focusing on use of type 1 pulinase as a debranching enzyme, and this deep branch linear glucan chains can be retrograded and for making resistant starch. For example, this is amylopectin, and this amylopectin here alpha 1,6 linkages are there, and this amylopectin is converted into amylose. And when amylose is subjected to retrogradation, it gives a crystalline and stable structure called as type 3 resistant starch. When it is complexed with some fatty acid molecules, it results into a crystalline and stable structure. It is called as type 5 resistant starch. So likewise, these amylose linear glucan chains can be converted into crystalline and stable structures, and it is called as uh, type 3 or type 5 resistant starch. So if fatty acid molecules are complex, it is called as type 5 resistant starch. When it is retrograded as such, it is called as type 3 resistant starch. So what our laboratory did, again, we actually explored one Himalayan thermal spring and we came up with a novel gene and we expressed it, and we found that we discovered a type 1 pulinase, which was a cold active. It was even active at 20 degrees Celsius, 10 degrees Celsius temperature, and it was a cold active enzyme. It shows remarkable tolerance at 40 degrees Celsius, which is a desirable temperature for making for hydrolysis of alpha 1 6 bonds and making this amylopectin to amylose. So from potato we extracted starch. So this method was already available. The potato starch is also available on the market, but we tried on Indian potatoes, and we thought that yes, we successfully extracted starch from potatoes, and this starch was subjected to type 1 pulinase digestion, and it resulted into amylose chain. It was retrograded, and we successfully generated resistant starch, and about 45 gram of resistant starch could be obtained from 100 gram of potato starch, so about 45%. And this was a scanning micrograph. So how these are the starch granules of potato. When it was gelatinized, it gives this kind of structure. Then it was debranched and resistant starch. So like this is a granular starch granules. This is a cartoon of starch granules. When it was gelatinized, it becomes like this. Then it was linear glucan chains by action of type 1 pulinase enzyme, and then it was retrogradated to helical structure, and this helical structure was resistant to digestion, and its crystallinity was increased, its thermal stability of this starch was increased. And when we subjected to the intestinal enzyme digestion, you can see this digestion of resistant starch did not lead to generation of glucose molecules, and whereas the starch, the native starch, could generate a lot of digestible product like glucose in the reaction system. So it was a digestion resistant starch called a resistant starch. Then we realized that a lot of peel is being produced by um, potato industry. So though that peel can be grinded, and we develop a method for starch production from peel, and that peel, about from 1 kg of peel we could generate about 40 gram of starch, and that 40 gram of starch was converted into resistant starch. So then we successfully generated resistant starch, and also the digestible proportion of the starch was subjected to alpha amylase digestion, and we could generate malto-oligosaccharide from that. So those all analytical analysis confirmed production of resistant starch. Then type 5 resistant starch we could generate from corn starch available in the market, and this cornstarch was complexed. First, it was debranched by using the same type 1 pulinase enzyme, and then it was complexed with fatty acid, for example, lauric acid. And this is the procedure explained, that how we did gelatinization, then debranching, then fatty acid complexation, and this resistant starch 5 was made. This is a cartoon explaining that how this amylopectin was converted to amylose, then amylose was complexed with fatty acid molecules. And these are the analytical techniques which confirm the high crystallinity of this type 5 resistant starch and high stability of type 5 resistant starch, and yeah, reduced digestibility of this type 5 resistant starch was confirmed. Now, different fatty acid molecules, saturated and unsaturated fatty acid molecules, were tried, and we could see that saturated fatty acid with carbon chain like 10 to 11 were highly competent in generating a very complex and crystalline structure as compared to the unsaturated ones. So this is a comparison of the type 1 pulinase. And yes, we are not the first. Some other publications also we got from Indian scientists who has generated, who has worked on type 1 pulinase. But resistant starch, I found that only our group has generated uh, this process for production of type 5 resistant starch, type 3 resistant starch. Then we worked on one enzyme, dextrose sucrase, that was able to transform sucrose into digestion resistant gluco-oligosaccharide molecules, and in presence of some acceptor, for example, maltose or isomaltose. So therefore, it is called as isomaltogluco-oligosaccharides. In absence of this acceptor, it could generate a polymer of glucose called as dextrin. Again, it is a very high value molecule, dextrin. So thus, this dextrin sucrase from an Indian strain, Leuconostoc mesenterite, we could generate glucoligosaccharide. It also has a very good big market. IMOs have a big market. Then the same dextrin sucrase can also be used for production of 4-galactosyl cozy-biose. The cozy-biose or 4-galactosyl cozy-biose is a very good molecule, very high value molecule. It is a trisaccharide, and this one cozy-biose is a disaccharide having glucose units linked via alpha 1 2 bonds. So we could generate these high value molecules by using a mixture of table sugar, liquid way, jaggery liquid way, molasses liquid way, and these all type of feedstock could generate alpha galactosyl cozy-biose, and it was confirmed. This analytical technique confirmed production of alpha galactosyl cozy-biose. Then we also worked on leuconostatic, Levin sucrase enzyme and Lewis sucrase enzyme can convert sucrose to leaven. Leaven is a polymer of fructose, and this leaven sucrase can also generate some prebiotic molecules like malubiols and manninotriose from raffinose and stachyose. So fructooligosaccharides are also desirable prebiotic molecules because its consumption actually flourish the gut microflora. Therefore, it is called as prebiotic molecule. And leaven polymer is also having several good functions. For example, one of our team member could generate a leaven-based film, and the mixture of leaven and green gum film we could generate, and it was a very good film, and we found this film useful because of having high elongation at break, high water solubility, high thermal stability. It can be used for wrapping and packaging purposes. So it was tested by different analytical technique, and we found this leaven-based biofilm useful in future purposes. Then some for example, banana, after harvesting banana fruit, the whole biomass, 80%, more than 80% biomass is waste. So the banana pseudostem can be extracted, and it banana pseudostem has a lot of medicinal property, and our Ayurveda has made a lot of importance of banana pseudostem. In south region it is useful, but in north part it becomes a waste probably. So we can generate a pseudostem extract, and these enzyme, dextrin sucrase enzyme, can be used for transformation of the sucrose present in it, and it can become a oligosaccharide rich juice. So it can become a functional juice. So that we have tested and tried, and we found that this functional juice was able to induce the growth of some probiotic strains in vitro conditions. Likewise, the sugarcane juice or sweet sorghum stalk juice, the sucrose present in it can be transformed into oligosaccharide by treating it with leuconostoc mesenteride dextrose sucrase enzyme, and we have seen that this sucrose could be converted into oligosaccharides molecules, which can induce the growth of probiotic molecules. So it is a functional juice, means having a function of increasing the gut microflora. So, and after extraction of juice, the bagasse which is present, from the bagasse we could extract xylem, and for that xylem can be treated by xylanase enzyme. So our team again searched for xylanase from our metagenomic data resource, and we can actually we reported a xylanase which was GS10 family xylanase, and it was highly stable gynase, and that it was having high salt tolerance, also high tolerance to alkaline acidic condition also. So this xylanase can be useful for converting xylene into xylo-oligosaccharide. That we have demonstrated that the xylene extracted from sweet sorghum bagasse can be treated with this enzyme xylanase, and xylo-oligosaccharide can be produced. Likewise, our metagenomic data resource was mined by one of our team member, and we actually characterized one novel gene which was encoding GH5 family endoglucanase that could transform cellulose into cello-oligosaccharides. Again, these are emerging prebiotics and they can influence growth of probiotics, therefore they are called as prebiotics. Again, these enzymes are showing remarkable heat and salinity tolerance. They were not only this, they were tolerant to detergent and organic solvents, which is a which is an industrial demand for their use in bioethanol industry. This is a comparison of endoglucanases available in some of good version of DLNOS tryptase, and we compared it with our DLNOS, and we found that it is substantially good variant of endoglucanase. Likewise, our team uh, work on inventing one or identifying one gene which was encoding glucose tolerant glucosidase. So glucosidase is important. A lot of glucosidase are available that, but they are not either they are not glucose tolerant or they are not thermostable. But here, this version of enzyme was not only showing a substantially high glucose tolerance, substantially good ethanol tolerance, but also its heat stability profile was good. So combining these three characters together as was a beauty of this invention. So it was having considerable heat stability, it was having considerable glucose and ethanol tolerance, which is a demand for this enzyme in ethanol industry. So this kind of invention was made by our group. So this is a comparison of different glucose tolerant glucosidases, and you can see that glucose tolerant glucosidases, the half-life of our version of enzyme was showing a very good half-life along with very good uh, relative activity in presence of glucose and in presence of ethanol. So then the other biomass was pectin. You know, there are carbohydrate, two major carbohydrate polymers are cellulose and cellulose and xylene. So we worked on xylene utilization into xylo oligosaccharides, cellulose utilized in cellulose oligosaccharides. Then pectin was left. So we mined our metagenomic data resource, which we generated by from Himalayan biosphere, and we could identify one endopolygalacturase variant, which was a acid active enzyme. It was active at acidic pH, and it was successfully generating pectic oligosaccharides uh, from pectin. So this was a very good invention made, CFTI also has invented one endoparaglutinase, but our endoparaglutinase showed better uh, biochemical profile when it was compared from the available data in literature about these endopolyglutinase. We found that our enzyme was showing a better half-life, a better catalytic activity for generating pectic oligosaccharides. Then pectin is an undesirable component in case of textile industry when they make high quality fibers for making cloth. So in textile industry, so therefore the pectate layers are important, but those pectate layers should be alkaline tolerant. So these alkaline tolerant pectins layers are important because pectin is soluble in alkaline solution. So by industrial processing of textile in textile industry, the plant fibers processing generate alkaline environment. So therefore, alkaline tolerant pectate layers are very much important. So our team invented one version of pectate layers which was alkaline tolerant, and we have shown that it can be used for bioscoring. Bioscoring means removing of pectin from plant fibers. So it was a very good candidate enzyme for bioscoring purposes, and it is useful for textile industry. That's it. I think this slide shows that how these enzyme consortia or a portfolio of enzyme our group has generated that can be used for transformation of stock juice, street sorghum stock juice or sugarcane stock juice into functional juice, but the bagasse can be used for extraction of cellulose, xylem, pectin, and these enzymes can be used for transformation of cellulose to cell oligosaccharide, xylem to xylooligosaccharide, pectin to pectic oligosaccharide, and pectate lyase is useful for bioscoring applications, that is removal of pectin from the cellulose fibers. That's it. I think it's time to thank my hardworking team. So this all work has generated six PhDs, and these PhD scholars they are placed in different countries, I would say. So and one of my student has got scientist position at a very premium institute of CSIR IICT. My post the NPDFs worked under me. They have got permanent positions in different government institutions also. My student Satya, he got awarded by this the honorable minister, and also Mashalkar Saab can be seen here in the picture. He got Gandhian Young Technological Innovation Award. He got MI Young Scientist Award, UCBT Award. So the work was appreciated by conferring fellowship from National Academy of Agriculture Sciences, INSA, Tata Innovation Fellowship, National Academy of Sciences. Some BRSI has also ISCA, Indian Science Congress Association has given Hiral Chakravarti Award. So these are thanks to the funding agencies, mainly DBT, SERB, now it is called as ANRF, the previous institute CIAB, NABI, as well as current institute GBU. Thank you very much. And yes, this is tribute to Mahatma Gandhi ji. Thank you all. If you have any questions, I'll be happy to answer.
Thank you, Professor Sudhir, for this wonderful presentation. Coincidentally, we both were in North. You were key innovators among them, and I was the technology transfer facilitator.
Yeah.
Now again, we are in the same city, adjoining GBU, State University,
Yeah.
under DST, and KSU, under DSD, same government umbrella.
Yeah, same government.
So hope your innovation and our skill will go hand to hand, and will keep on providing good platform to young innovators,
Yeah.
entrepreneur through this kind of platform. Thank you so much for the wonderful presentation. We have questions here. Dr. Sanjeev Kalita is asking whether initiative for upscaling toward industrial production in a downstream process are also taken, especially in case of tuberose. So can you just highlight those points?
Yeah,
Upscaling.
means previously being a laboratory scientist, my the demand of my job was to do innovations. So our team did innovations. We did some academic research also. We did innovations, and we were looking for industrial partnership for its upscaling. But yes, now because I have shifted to GBU and it has capacity for upscaling, so I'm planning for to take up this challenge of upscaling because I have learned that industry they have so many things in their mind. I don't know. They take my innovations, and then something is stuck. In this collaborative research is not so. I should not discourage people. So means we have to improve ourselves basically. So therefore, I'm planning for upscaling. As of now, I have done means my team has done innovations, and some of those innovations should be taken up. So I was trying to collaborate with industry, and I have collaborated. And recently, one very good industry of Ambani Group company, they have taken some of our patents, and they are saying that they will translate, but when I don't know. So so therefore I am now in the field, and I will see that by collaborating with my colleague in my department how I can take up this challenge for upscaling and downstream processing. Thank you. It is important point, valid point raised by you.
Yeah, thank you so much. And since this is a agri biotech program, agri innovations, and largely it is surrounding biotechnology. So today morning itself, I humbly requested your Vice Chancellor, Professor Majumdar.
Yeah.
He kindly agreed to join our valedictory. So
Oh,
that will
that's it.
commence at 7:30 to 8. So the
Oh,
same link can be used. If your time permits, please join us also. And just for the information of audience, we'll take a break. And as per program schedule, again we'll start by 6. 6 to 7:30, we'll have final presentation from the industry, the floriculture part, orchid and floriculture industry perspective. Then we'll have an elective program for 30 minutes and followed by the very mandatory, that is a kind of assignment. There will be 25 questions, whatever we have discussed in last 6 days. It is in multiple choice question, MCQ, no minus marking, and each question will have 4 marks, so total 100 marks, 25 questions. So whatever we have discussed, all those things will be the part of assignment. Simple question, not in very critical, but at the same time, it will assess that how much attentive we were there during the program. So I think Dr. Susmita Shukla has raised her hand. So uh, admin
Yeah,
can please
he has
unmute.
animated.
Yeah.
Thank you so much. And Dr. Sudhir, it is a wonderful part of your work which you have shown and showcased in this particular FDP. Really will be very good and information for all the scholars and the faculties who are there. In our department, we too have downstream processing unit actually, the students who are learning and teaching, but that too in lab scale. So in GBO, is there anything which can be done for student internship and all? Is there any facilities over there where student can come to you and they can be trained for such kind of thing, particularly for your work, the work which you are doing?
yeah. At present, we have a good number of MSc students, and they all we undergone MSc dissertation program also. We have PhD scholars also. Outside, it depends upon the faculty interest. If they can, two faculty of different institution can collaborate and can work, definitely. There are procedures for that. But yes, we are developing means different facilities and very good equipment resource we have generated now, and we have developed some good facilities, including like fermenters we have procured. I think a couple of months ago only one fermenter of 50-liter arrived here, and it is under installation. But yes, a small 5-liter fermenters are running at present. My students only run those fermenters. So those things are under development, and yes, there are procedure for everything. Yeah.
Okay,
We can
thank you so
discuss
much.
after the program. You can just write to me. I will facilitate the collaboration part and whatever discussion that can take place, we'll take it up further after this program.
Okay, fine. One more thing I want to ask if you permit.
Yeah, please go ahead. Time is there.
So only the thing is that we are having a mandate, whatever dissertation part the students are going on, they need to come out in form of their outcome, means in form of one paper or publish the paper in Scopus indexed or peer reviewed journal. So in some of the institute, what happens, people go work there and they don't allow them to write or publish paper in collaboration. So that too, we need to find out the feasibility. That type of restriction shouldn't be there, which I think. So is there in that case, KSU and GBU are having this kind of thing, so that can we come together and we can go for
It depends on the IP policy. Very much I was dealing with this matter in the past also. Depends on the IP policy, what is to be disclosed, what is to be published. So I think that can be discussed separately. That is part of larger collaboration. And we can take it up, whatever rules, regulation, and the IP policy basically permits, it will go according to that. Always it will be student and faculty friendly, no doubt about that, being a public university. So we'll take it up in due course. Just write to me.
Thank you so much.
One question we have from Dr. Pompey Das: how to define quality assurance, quality control parameter for prebiotic enriched functional juice towards translation?
So, quality control means in our case, analytical techniques have proven that yes, um, it has the prebiotic oligosaccharide, and by using the transformed juice as well as untransformed juice in vitro condition, we have tested that the transformed juice can increase the stimulate the growth of probiotic strains. For any approval, yes, biosafety analysis and everything can be done, but we have not used any pathogenic microorganism. We have used the grass status strain for it, so there should not be any problem for that.
Another question Arun Chopra is asking, with the rise in temperature, can the thermostable enzyme address abiotic stress in the plant?
So, this is a good question. There are several metabolites, and I am working on it. I have not shown those things. So, I am collaborating with the very great plant institute of country, NIPGR, and some of our molecules like allulose and trehalose, they can be used as a priming molecule. So, if we a very low concentration of these rare sugar molecules, because it is a natural molecule, so it is better than the fertilizers or pesticides. So we have realized we have results, but I have not shown because it is means IP policy doesn't allow it because we have not yet protected the IP. So when we use a very minimum trace amount of these metabolites, microbial metabolites, we could see that it gives stress tolerance, including dot tolerance, including biotic tolerance to the plant like rice. So, for example, rice, there are blight diseases, and blight disease causes disaster for rice. In fact, the fungal blight, there is very minimum control is available. But this our method has means we are finding we found very good result, very promising result in terms of controlling those the fungal based blight disease. So this is a very valid point raised by you. So this increases the demand of these kind of molecules. Actually, the next era is for these prebiotic and rare sugar molecules, because we are not only facing human health problems associated with high sugar doses in our food products, but also our agricultural crops are facing challenges, environmental challenges. So we need to come up with innovations for to withstand these kind of challenges.
Great. Arun Chopra is writing something else. Like Rubisco, largest expressed in universe, can photosynthesis be increased? Arun Chopra ji, can you just unmute yourself if you want to talk?
Means those is not in my domain to comment on Rubisco, but yes, scientists can do, means they can, if they can change C3 to C4 plant. So then in future this can be done, I believe. But this is not my domain.
Thank you so much. So I don't think there is further questions. And for the researcher, for academia, it was very wonderful presentation. And I know that how much important it is, because in the past we have been trying to bring industry collaborators. Already we had generated a good lead. Hope we'll have a new industry partner for this functional sugar, tyrannose, and very, very, very important enzymes for the, especially in context of biomanufacturing.
Yeah.
Bio3 policy, it becomes highly, highly relevant in your job. Thank you so much.
Namaskar.
You all the best.
Thank
Namaskar.
you. Namaskar.
Dear participants, the final session is very, very important in terms of the certification part. Your presence in the valedictory, group photograph, feedback. Feedback, I would repeat, it is a today to twice feedback. One feedback for the attendance purpose, the way we are doing regularly in the Google form, and the other feedback you will be getting time till tomorrow also in your AICT login. I hope you have not missed track of your AICT login where you have submitted your application. I know it was a tedious process to submit application, upload NOC, your ID card. You should log in on the same Atal portal for submitting feedback. You will be getting option for the submitting feedback, and that feedback will be submitted to the AICT Training Learning Academy, Atal Academy. So I would say three components for the final thing. One is a attendance part, what we have been talking, 70% attendance. Then a score of the assignment. And third is submission of your feedback form to the AICT portal. Once these three aspects will be completed, then you will be getting certificate for this entire FDP, which is highly relevant for your API, for documentation purpose, and for further reference. So again, we'll meet at 6. Then we'll start one session, followed by valedictory and assessment. So have a break and see you again. Thank you so much.